Thanks very much, Will, for the invitation to be here, and thanks all of you for joining us tonight. It's quite an honor to be here in London, here at the Linnean Society, under the watchful gaze of the man himself, and here actually with the Systematics Association. So I feel right at home with all of you systematists. So today, what I'm going to do is tell you a little bit about my vision of a future for fungi. And it's from, of course, my perspective of doing research on lichens at the Royal Botanic Garden in Edinburgh. And you might all recognize fungi getting a little bit of a bad rap. And does that ring any bells for you? In literature, it's all about moldering and decay and death, and it's all a bit gloomy. And even our prestigious scientific journals instill a fear of fungi and tell us to fight fungi. It's all a bit grim. But actually, I don't know about you, this time of year, fungi are springing up all over the place, and not only in the fields and forests, but also in my lab, in the metro, in popular accounts. So that's a kids magazine from RSPB, in The Guardian, on TV shows. There's an IMAX movie coming out. Fungi are experiencing a real moment right now. It's not limited to the United Kingdom. This is a bookshop in Switzerland where I was last week. It's all about fungi all around the world. And it's great. There's a real riding the wave feeling. Thankfully, some of the scientific journals have picked up this, and it's a bit of a more positive spin. So this article is about some work undertaken by these two, Merlin Sheldrake and Toby Kears. Merlin Sheldrake you may have heard of because he's written this book, Entangled Life. If you've not heard of it, it's a popular account of how fungi work, just how alien and bizarre their life histories really are. And he's done a great job bringing that to a much wider audience. Toby is a very talented experimental biologist and is now working with a team on, I love this, they have created the Society for the Protection of Underground Networks, and they are a network protecting networks. So it's all changing. And I think right now there is riding this wave of public interest, but also thinking about fungal conservation. Because who wants to conserve something that's a nasty parasite? Nobody. But if people start to appreciate how important they are in underground networks and all the other roles that they perform for us in the world, then it becomes a more viable and a more interesting activity. So the talk tonight is in 3 parts, and I'm going to start with a little fungi fest. I'm not sure how many of you are interested in fungi. Hands, show of hands. Yay! Do we have any active field workers in fungi in the audience? Researchers? Just enthusiasts. Yeah, I love it. Okay, fantastic. So we're going to do a very brief fungi fest, and then I'm going to tell you about some trends from the research of the Botanic Garden. And I'm going to have 3 different time frames for that. So looking back to an archaeolichenology project, there's a word you've never heard before, through some present research, and then looking to the future as well. And I'll end with something about global conservation. And I think something went wrong in my biography. I don't think it was your fault, Will. I think that might have come from Wikipedia. Goodness knows who did that. But I've only been the co-chair of the IUCN specialist group for a few years now, 3 or 4 years, so not since 2010. So it's relatively new for me, but quite exciting, and I'll tell you my views of that as well. So let's think about fungi. You probably all recognize them. Sounds like we've got a lot of enthusiasts in the room already, which is great. You recognize them as the recyclers of a forest, as maybe from your fruit bowl with little growths like this. They're the nitrogen fixers in your lawn. They're symbiotic organisms with trees, and they're inspiring for their beauty. So probably a lot of people get interested in them just because they're gorgeous. Lots of inspirational artwork is going on with fungi these days. Their life histories are just as diverse as their sizes and shapes, and they do all kinds of crazy things, from living on and in jet fuel to living in anaerobic guts of rumen animals, rumen-containing animals, to the sort of cryptic diversity that we see under the soil and in water. And those are sort of the most diverse places we find them. So oftentimes they're hidden, and then they sometimes spring up magically and inspire. They reproduce by spores. You probably all know this, microscopic tiny particles that get lofted up on the wind. And they have come up with a myriad ways of increasing surface area to make more spores. So from gills to wrinkles to tubes to pores. They can make sexual spores like this, or asexual spores indeed. They can use clonal reproduction, making just fragments of their bodies that disperse, and sometimes they'll carry their symbiotic partners with them when they do that. So they're they have a million ways and that probably is a million ways. I'll tell you more about that in a minute, of dispersing. And here they even use animals for dispersal. So this is the zombie ant. Hands up. Zombie ants, yeah, nodding. You've probably seen the TV show, right? So the fungi infect the animal climbs up, the fungus emerges when the animal is up high to disperse the spores. But of course, fungi aren't all about beauty and evolutionary innovation and biodiversity. They're also a bit about the bottom line. So this is the single most valuable fungus in the world. This is the brewer's yeast, Saccharomyces cerevisiae. And this single figure, a financial figure, is quite an impressive one. Single fungus responsible for 5% of the global GDP in the United States. 3% of the US workforce is involved with this single fungus. So it's an absolute workforce. It's involved in brewing, it's involved in baking, winemaking, yeast-derived insulin, you name it. So it's a massive input into economy. And that's one tiny microscopic example of the value, the economic value of fungi to us. In this very room a few years back, I heard a whole talk about the economic value of fungi, and I was totally blown away. So this is just a tiny tip of the iceberg. And if you're interested in yeasts, there's a fantastic book by Nicholas Money called The Rise of Yeast. Pretty clever. This is from a review paper, and it's all about how people use fungi. This figure is all about the enzymes that are used by people, that are produced by fungi. And you might see blue jeans in there, and you might see wine, and you might see biofuel. There's all these different things, and the paper is kind of funny. They harken back to the 50 Ways to Leave Your Lover by Paul Simon a long time ago, right? It's the amazing potential 50 ways we can exploit fungi. So here's some of the uses against diseases, human diseases, plant diseases, enhancing crops and forestry, food and beverages, and even slightly aspirationally saving the planet. So one of the things that they highlight, of course, is the ability of some fungi to recycle plastic for us. So that would be great, and there's active research there. So coming to a recycling center near you, your local fungus. Now, what about the diversity? I've mentioned about biodiversity. My title is Lichen Biodiversity Scientist. I'm naturally interested in that. The biodiversity of fungi on Earth is a bit of a black box. That's not a mistake. That's a big black box, because actually we talk about the missing fungi. We know they're there. We can sense them by their sequences, but we don't know what they are. That little tiny corner up here, that yellow corner, is 3%. We estimate that we know about 3% of the world's fungi, and the rest of those estimated fungi are unseen, and we know about them only from sensing them from their DNA. So that's pretty interesting. Any ideas about how many fungi are out there in the world? There's a growing scientific consensus about this number. Orders of magnitude? 2.5 million. 2.5 million, that's right. That's the answer. Right now, we've all converged on that kind of 2.5 million is where we think we are. Exactly. And that figure surprises quite a lot of people. So thank you for spoiling that, my fun. I was hoping somebody would say 10 million, 3,000. Yeah, 2.5 million. It's a big number. Okay, so let's move away from that amazing diversity of fungi and think about a smaller group of fungi that I study, the lichen fungi. And I'm going to tell you about some trends that we've been looking into using lichens in the research of the Botanic Garden. And the first trend I'm going to talk about is looking at biodiversity loss in that transition from pre-industrial period to the post-industrial period. And the way I'm going to do that is by using lichens. But what are lichens? Of course, they're symbiotic fungi. They have an ability to form protective structures just with these fine microscopic fungal threads called hyphae. They can build these fantastic structures. This is a cross-section of this lichen. And all that you can see here is fungal tissue except for that green layer of algal cells. They make a ton of diverse chemicals which can protect and promote their algae or their bacteria. So this little zone at the top that's kind of pinkish brown is a chemical that is deposited only there as a UV screen for their algae. And the black is melanin, just like we have melanin in our skin. They use melanin for protection in a similar way. So they form these complicated structures. They're out there as symbionts in the world, making new biomass for creatures to eat. And they obtain their mineral nutrients from the atmosphere. So we know they get their carbon from their symbiont, but all their mineral nutrients and their water they get from direct deposition onto their surfaces, which is great, unless the stuff that's directly deposited onto them is full of toxins, which sometimes in the world it now is. So that's just a little background of lichens. Now we're going to think about the past. So I'm going to start with pre-industrial Britain, in fact, medieval Britain, just as a little thought experiment. What was going on a long time ago here? Not a heck of a lot, I would actually argue. So there weren't very many people. They mostly lived on the land. They did not live mostly in cities. They undertook activities like this, beating acorns out of trees for their pigs. They coppiced the local woodlands to produce fodder and building materials. They mostly had a relatively low impact on the land. So at that point in time, the fungi and the people sort of managed to coexist probably fairly well. Move through time and you get more people, more land use, less habitat, and more heavy impacts. And eventually you end up with a situation here in London in 1952 with the Great Smog. And that single event, probably you all know, was really deadly. It killed between 4,000 to 12,000 people on that single day. That was a really critical day. And that's just emblematic of the kind of landscape changes that we've seen in this over this period, crossing that threshold to industrialization. So in thinking about what that means for biodiversity, here's a little timeline going from somewhere here now back in time. And let's put some things on that timeline. And the first thing I want to put on there is quite apropos because we have Mr. Linnaeus staring down at us, is the publication of Systemae Naturae back in 1735 when he started publishing these volumes. And that marks the beginning of the activity that we call systematics. He did the first job of systematically putting together an arrangement that other people could follow so that we could build taxonomies for lots of different species groups. And that was a really important moment in time, and it allowed the activity and the school of taxonomy and systematics to develop. So this is the number of taxonomists and the outputs of taxonomists over time from 1750 to, I think, somewhere around 2000. So even though we sometimes decry the lack of taxonomists, and I know that's a real concern, our outputs somehow have still been increasing and more people are involved in it. So that's positive. But unfortunately, the publication of this and the beginning of this activity of taxonomy and systematics coincided almost precisely with industrialization in the big cities in Europe. So in London, we think it was about 1750 when that sort of threshold of industrialization was crossed, along with all those landscape changes that happened at that time. So that's a pretty unfortunate coincidence. Just as we're starting to figure out how to explain and describe all this diversity, the impacts of it are becoming rather severe. And of course, the impacts of industrialization are many. Those words are going to be way too small for you to read. But this is from the Living Planet Index from the World Wildlife Fund, looking at all the increases in things like building dams and forestry, human population increases, marine fisheries increasing, materials extraction. You can imagine all the bad impacts that humans have had with this rise of population and loss of habitat, resulting in a loss of wildlife measuring 70%. So I'm not sure if you've seen that report, but the Living Planet Index tracks species from 1970 through the present. This is really hard. This little wobbly thing doesn't go straight, sorry. So from 1970 to 2018, that's a 69% decline of 5,000 species and 30,000 populations that have been tracked. So this is a massive decline. And in recognition of that, of course, the Convention on Biological Diversity was signed in 1992 by all the civilized nations on Earth. As an American, I think I'm allowed to say that. And that recognizes the decline and looks to try to stop and reverse those declines. Now we have a bit of a problem, don't we? Because when we have the beginning of this activity of describing the nature of the world and the huge impacts of industrialization, it makes us wonder, what are we missing? What did we miss, basically? And we're pretty lucky in that lichens are a pretty untapped archaeological resource. This is the result of a conversation that Brian Coppins had with an archaeologist who showed up at a meeting with a bundle of wattles under their arm. And she said, "What do you make of these?" And he said, "Oh, it's quite interesting. Where did you get those?" When the conversation developed, she said one thing and he said, "Well, that's not where they grow." And that idea developed. Chris wrote a grant about this, and the three of us worked on this project for a few years. And it's based on two observations. One, that lichens grow on bark. They grow on bark. When you cut down a tree, the bark stays on the tree and the lichens can be preserved on that bark. And number two, trees and other wooden substrates are used to make vernacular buildings, these sort of low status places like thatched cottages and pubs and other low status buildings. During the project, I talked to a thatcher and he said, these buildings were built from the things you could see from the ridgeline of your roof. That's key. It means that all the wood that was in these houses was collected right around there. So you're looking at individual herbarium collections potentially in every house that's still standing. So here's what the fieldwork was like. Go and find some pubs, have some cups of tea. It was lovely until you had to go up into the attic filled with dust and insulation fibers. We had a lot of buy-in from the people who owned the buildings. We had one person say, come back tomorrow, we'll see if we can get you access. And indeed, they tore a hole in their ceiling in their living room so we could get up there and have a look. That was quite surprising. The buildings experts that we consulted about this to try to find and understand and how to date the buildings said, you're probably not going to find any bark in there. So just be, don't get your hopes up. Well, the truth was we found lots of bark. In over 100 buildings, we found preserved bark with preserved lichens. Over 100 species of lichens were preserved on the bark. The preservation is pretty good. Can anybody recognize that one? Yellowish leafy lichen, come on, yeah. So this is the what do they call it? The sunburst lichen or the yellow wall lichen, Xanthoria parietina. And so the preservation was better than we expected. It wasn't always easy. So this is what a historic sample looked like in one case, and this is what the modern material should look like, quite different. But we're lucky that the spores and chemical reactions were preserved, and very lucky that Brian Coppins was there, who's the preeminent lichenologist in Britain who can identify pretty much anything. So he was very helpful with this process. And we found lots of interesting species. So here's one of 11 species found outside their modern ranges. So this is Caloplaca ferruginea. You can recognize it by those kind of rusty dots on bark. It was found here in Cambridgeshire from 17th century materials. You can see that the modern dot map of this, which is all these black dots, are well away from that historic locality. And the idea here looks like, well, I think maybe the temperature could be a difference here. So the driver of the loss of this species from the current landscape could be temperature. So let's look at the 95% confidence interval for the current distribution here, hovering around 7.5 degrees. And there's the village where the historic material was from, looking at about 9.5 degrees. So that's what a 2-degree difference looks like for the species. So this species can't grow here anymore, it's too hot. And in 17th century Britain, that was the middle of the Little Ice Age. So it confirms the kind of patterns that we expect. So some species have certainly shifted their distributions. You wouldn't expect that one there today. Here's another example. This is Protoceria liaplaca with massive big spores. These are 40 microns or up to 100 microns. So they're pretty easy to identify by spore, even if the material looks a bit rough. And this dot map is a very typical dot map for a species sensitive to pollution. You can see that the industrial north and around London, there's no dots. So this is the modern distribution it could be anywhere except where the pollution is high. So if you look at the 95% confidence interval for sulfur dioxide tolerance of the current distribution of the species, it's down here around 300 parts per billion. And those villages in Essex are out here at 700 and 800 parts per billion. So this is a clear signature of a pollution intolerant species. So we could do that. We could look at every single species one by one. Yeah, local losses. But we decided we really wanted to look at the community level and try to figure out overall what was happening. And the idea is to then compare our historic lichens that were present in the historic landscape with the present day, right? And so if there's something present today, and it was present a long time ago, there's no change. That's not particularly interesting. But if it's present in the historic record and absent today, then we think we can infer a loss. Obviously, if we couldn't see it in the historic sample, there's a lot of reasons we couldn't have, that we didn't see it. Maybe it wasn't there. Maybe we didn't collect it. Maybe it wasn't preserved. There's a lot of reasons it's an intractable problem. So we're going to focus on loss. How much loss can we infer? And how can we be confident that it actually is absent from the modern landscape? And this is where Chris Ellis's expertise comes in. He's very good at statistical modeling and using the British Lichen Society record data for for the distribution of these species. We looked at our target sites and the distribution, the modern distribution of lichens around them, and established statistically robust thresholds for what would be present and what would be absent. And then we're able to infer losses across the entire data set. We had 3 villages that we sort of focused on, one in Somerset, one in Wiltshire, and one in Essex. And those black parts of the pie show how much biodiversity we estimate has been lost across that industrial threshold. It's huge. So the smallest losses are 35%, the largest are 80%. Imagine going to your local park and 80% of the species are gone. It's a huge change. And I think some people who work with sort of indicators of pollution would be expecting something like that. But I think at the same time, that number is sort of surprisingly huge. So what can account for these losses then? So if we look at this site here in Essex with the highest losses, we know already that Essex is just too close to London from a pollution point of view. And if you look at the sulfur dioxide context of Britain, overall, the average sulfur dioxide concentrations are here. Somerset and Wiltshire sort of sit within that distribution, but Essex is well above it. So we can certainly suggest that the highest losses correspond to a pollution problem. And then we want to look at this one down in Wiltshire. So we've got much less loss here. We looked at a few things, not just pollution, but also the extent of ancient woodland and also climate. So this is a story about the extent of ancient woodland. So the extent of ancient woodland in the Wiltshire landscape is really quite large. So this is percent ancient woodland in the current landscape. And overall, it's ridiculously low. I can't remember what the numbers are. It's less than 1% overall. But Wiltshire has 2.8, nearly 3% of ancient woodland still remaining. And we think that that offers a buffering effect from all these other negative consequences of pollution and whatever other land use changes had happened. So we're able to sort of infer what those drivers of loss are. And those numbers between 35 and 80% really accord very well with those numbers from the Living Planet Index, don't they? Of 69%. But what we've done now is broken up that Living Planet Index trend by geography. So in North America and in Europe, if you look at those trends, there's still a decline, but it's only around 20%. Whereas in the global south, where industrialization was much later, the current declines are much, much steeper, from 50 to 90%. So the declines that are happening now in current times in this global south, they happened before here. It's not that there weren't any declines, it's that they're before the baseline of 1970. This is the problem of shifting baselines. It's a well-known problem, but it's important to consider that those big declines that we saw of 80% happened way before this period, didn't they? The one other thing I want to sort of bring into this conversation about the Living Planet Index, as robust as it is, 5,000 species is no mean feat, but all those 5,000 species have backbones. So we've got good trend data for lots of things if they're relatively straightforward to monitor. You can count them, you can see them all year round. You've got a big bunch of public interest and investment in it. So for things like lichens, that's not as easy. What's great is there are big data sets. So the British Lichen Society has been collecting record data for, well, going back to the 1600s, but the more modern mapping data goes back to the 1960s. So we've got big data sets, 2 million records now, opportunistically sampled. So not a systematic sample, but still lots of data. And there's a group down at the Center for Ecology and Hydrology, Nick Isaac's lab. This is a paper by Charlotte Elfwaite and co-authors looking at trends. These are occupancy models for different smaller, harder to study organism groups. So it's freshwater in pink. Those are freshwater invertebrates, insects, invertebrates that are not insects, which is basically spiders. And then we've got bryophytes and lichens in purple. So let's look at that bryophyte and lichen trend here in purple, climbing and climbing and climbing from 1970. That's good news, isn't it? It's looking good. These are the kind of models that were used for the State of Nature report. So some of you may have seen that report. And the overall bottom line is it looks like a recovery since 1970. Of course, when you're thinking about that, sorry, it's not going. Go. A single trend line like that hides a lot of variability. There's 1,300 species in that lichen and bryophyte data set. So if you break it down a little bit, which they started to do, you can see lichens in blue here was a huge recovery, apparently. Bryophytes, not so much. And then common and rare species. So the common species are increasing, whereas the rare ones much less so. Still, that's quite coarse analysis, and it's worth looking more at that. I've already told you that there are a lot of species of lichens that are sensitive to pollution. So when we're thinking about this recovery, the potential reasons for the recovery actually are quite clear in some ways. This is a map of Britain that was published by Oliver Gilbert in 1974, and it was made by 15,000 school children who went out to their schoolyard and counted the number of lichens that they could see, the different species. The areas in black have no lichens at all. 1970, no lichens. I asked my predecessor at the Botanic Garden, who was an inveterate list maker, why is there no list for the Botanic Gardens? He started his job in 1970. There were no lichens in the Botanic Garden in 1970. Now there are probably pushing 100 species there. But at this time in the 1970s, there were none in vast areas of the UK and other European places as well. And that mirrored very precisely the concentrations of sulfur dioxide. So the purple areas have the highest concentrations of sulfur dioxide. That's from 2000. So even in 2000, those concentrations were quite high. And the mirror is quite remarkable, even just down to, you can see a spot for Grange mouse there, which is even visible. So that's interesting. This area in gray, where there's a single lichen, I hope you can discern there's a gray patch around all the dark black. There's a single lichen there. It's an acid lover, this one, which is incredibly rare in the landscape now, and they're thinking of red listing it in the Netherlands. Hands up if you know what red listing is. A little bit. Okay, I'll talk more about that later. So we have a problem where we have lots of areas in Britain that had no lichens at all. And it's not just lichens that suffer. 33,000 people are suffering premature deaths every year, even now, due to air pollution. So this is sulfur dioxide. It's a very damaging pollutant, not just for lichens and bryophytes and other critters, but also for people. And as a result of that, the clean air legislation was passed. Now the picture is looking a little better. The measuring is slightly different, but the glaring bright colors of high concentrations of sulfur dioxide in this image are gone. So because of all that regulation, sulfur dioxide limits concentrations have dropped right off. So that's great. That is probably a big part of that lichen recovery that we've seen. Lots of things that were killed by sulfur dioxide now can come back. However, this is a map of nitrous oxide. So we get some bright colors in here. And we're getting a little bit of high concentration of nitrous oxide. Those are pollutants that come, like sulfur dioxide, from burning fossil fuels. There's no safe limit for nitrous oxides for life. They're very dangerous for people. So they're a bit of a worry, and they're regulated to, everyone's trying to keep them lower all the time. But it's not just nitrous oxides that are increasing, it's also ammonia. So this map shows ammonia concentrations across the UK, and you can see how high they are. So up to, it doesn't matter what the values are because you won't have a reference point for it. But the point of this is that ammonia pollution is extensive. The values on the right-hand side here, my typeface has changed, are the thing to focus on. So 87% of England's land area exceeds critical thresholds for ammonia for sensitive species. That means sensitive species of lichens and bryophytes die because of those concentrations of ammonia across almost all of England, across almost all of Northern Ireland, across 60% of Wales and 20% of Scotland. So where there's a recovery from acid rain and sulfur dioxide, now we have a problem with nitrogen compounds. So if we look again at this trend of this purple trend of going up, let's unpick it a little bit more. And these are some of the individual species trends. These are occupancy models of individual species from that alphabet paper. And it's really interesting to look at them. Some of them have very low occupancy, rare species. Some of them are going down. Some of them don't fit the models very well, so you've got red dots. Some of them are going up. You can expect that I didn't choose all those randomly. I didn't choose them all randomly. Based on the work of Chris Ellis and colleagues at the Botanic Garden, we have epiphyte community indicator species. Species of lichens that indicate a particular kind of habitat. The ones on the left are late successional oligotrophic sites. So these are mature woodlands with low nutrient input. You can expect there's not a lot of those woodlands around, and the lichens are low occupancy. You've got a lot of rare species out there, and they're not doing great overall. Whereas the early successional and middle of the road nutrient situations, you get a lot more higher occupancy species and more increases. So it's quite interesting to see those models and start to unpick it. That's a very coarse analysis, but I think it's worth trying to look at the guilds of species and how they're responding to these changes. And I think it's important to understand that the past biodiversity loss, looking forward to recovery, is now recovering into an entirely new situation. So even those vast baselines that we have, they're not going to make sense in our current environment. So let's think about our current environment then. We're going to move into the mountains. And this is a little story about species discovery. The tops of the mountains in Scotland have some pretty high areas where snow used to lie late. It's doing less of that all the time. But you can see my little dotted line. I hope you can just see a dotted line. And that marks out the boundary of what we call a snowbed. On the outside of it, you can see the graminoid vegetation, which just can't survive in this inner zone because the growing season's too short. So in these snowbeds, it's dominated by lichens and bryophytes. They've not been particularly well researched because it's hard to get up there, frankly. It's a long drive, it's a long walk, and then you have to be a specialist on top of that. So you sort of narrow your field of people who can do the work. On a single, on a few different survey days, I think I was about 10 survey days I was up there. I brought back a chip of rock. You can see the size of it. That's a 20p coin. And on that single chip of rock, and that was just on a scouting day, we ended up with a species that was thought to be extinct and one that had only been seen twice before in Britain. So it was sort of like, okay, we need to do some more work up here. We have no idea what's up here. And I don't know if you can see those blue dots, but there's blue dots on Ben McDewey, on Cairngorm over here, on Benavert over here. So the more places that we looked, we found these species. So the inventory work is yielding new records and new understanding of what's out there, which is really useful. But we also wanted to do some barcoding of those things to try to look for hidden diversity. And so barcoding is a technique that uses, the idea is to take a single species and generate a single unique sequence, short read of a sequence that represents that species. So ideally, one species can be represented by one sequence type. And the idea of that then is to have expertly identified materials yielding these sequences, which you can then line up and put together in a reference database like the Barcode of Life data system. So the idea of a reference database then is so that people who are not experts in taxonomy and systematics can generate a sequence. Many more people can generate a sequence than can identify a lichen. So the idea is to populate that database. And the database has been populated to the tune of about 11 million records so far. And then if you have a query sequence, you can line it up with all those other things and see, does my sequence match over here? Or does my sequence potentially result in a new species that's not been seen before? So it's a way of allowing non-experts to access expertly identified material. And it's also a way of doing some hypothesis testing around what species boundaries are as well. So it's a process of reciprocal illumination. I have a species hypothesis based on my morphology. Can my sequences back it up? So we ended up looking at 6 different field sites up there in the snowbeds, and there's hundreds of samples. And I'm very pleased to say that we have now a lichen barcoding technician, Amanda Jones, who's been working with us for 9 months-ish. She's fantastic. She can take a little rock. This is about 2 centimeters square, and there's 4 lichens on that. So just do that for you, like do your thought experiment. 2 centimeters squares like that big. There's 4 lichens on that. You've got to be pretty expert to figure out which is which and get it in that tube so that you can get the DNA out and then generate that sequence. And she's been doing a fantastic job of that. And out of our first 67 sequences, which represent the first tranche from that one site, so we've got 23 that have a sequence match in those reference databases and 34 that don't. Laura probably doesn't even know this yet. So that is a high proportion of things that don't match. I think that's quite interesting. There's a lot of reasons why that might be. One of the reasons is like this species here, which is a Miraquitica. So in this column here, they're known species. We know they're out there, but they've just not been sequenced yet. So that's great. We're adding new knowledge and we're adding more things to those reference databases that didn't exist before. We could have things like Trapelia over here, where that's been recently monographed and there were sequences of everything, yet we have new sequences in Britain. So we think there's probably a new species there. And then we've got some that are unknown from the UK. So here for Stereocolon, we have species we have samples whose sequences match things only known from North America so far. So we've got a lot of novelty coming in from that single small set of sequences. It's really exciting to be able to think that we're adding knowledge to these big databases. And that's important because those big databases are being used on really large scale, ambitious kinds of projects like this. So the International Barcode of Life has a project called BioScan, and that project brings together 70 institutions from 30 different countries with the large global ambition here of looking to monitor planetary biodiversity. How do you monitor planetary biodiversity? By using bulk samples. So you sample the soil, you sample the air, you sample the water, and you look for those sequences and how many are there and what's the diversity of those things. But of course, that all depends on having all your specimens with good reference data in the first place. So populating those databases is an important part of my job and Laura's job. So here's that figure again, 69%, right? I've mentioned already that we have similar figures from our losses of lichens going forward into today's landscapes. I've mentioned already that these figures are only so far based on vertebrates, but now that we have these techniques, we can start adding trend data and be able to hopefully look at trend data like this for a project like BioScan for these harder to understand species just based on sequences. So that's the ambition. It's a big one and it's a bit long term, but we're moving towards it. So I find that quite hopeful. Now, the last story I want to tell you about is another trend in lichen research, which is about looking towards the future. So if we think about all the losses that we've had, our ability to monitor species into the future, and the losses of biodiversity, we can think of where that biodiversity is most concentrated. And for Britain, one of our most important biodiversity sort of conservation assets is the temperate rainforest. This is another kind of thing that's having a moment. Anybody hear the BBC piece earlier this week or last week on temperate rainforest? There was a great interview. It was on the news. There's a book about the temperate rainforests of Britain. There's the Alliance for the Scottish Rainforest, which brings together stakeholders who are managers and recorders and specialists, and to try to protect and conserve and then rebuild what's left of these things. So in the UK, we have remnants of rainforest. There's not a lot of it left. It's a tiny fraction of what it used to be, and they're quite separated. So the idea is to try to protect what we've got, get rid of the rhododendron ponticum, try to reconnect these fragments so that in the face of climate change, species can move through them. And in the face of big tree planting campaigns, we understand what to expect from the recovery of the species that depend on these habitats. So let's think about lichen diversity in the UK. There's about 2,000 species, a little over that. And this chart shows the conservation evaluation of UK lichens. It's a little out of date, 10 years or so old. And we've got the threat levels from threatened to near threatened on the top. So we've got some threatened species here. So a couple hundred, but looking at 400 threatened lichens. About half of them are least concerned. So that's good. We don't have to worry about their populations. And then we've got some extinct and some data deficient species. So the blue bars show the threat levels and the number of species at those levels. But I'm really more interested in the orange bars. And so those orange bars are our species of international responsibility. And what that means is that in Britain, we have what we think of populations, probably 10% or more of the European or global population of those species live in the UK. So we have the responsibility to conserve them because most of their populations could be with us. And so even for the least concerned species, we've got quite a lot of international responsibility to conserve them. So they might be a weed for us, but I was in Switzerland last week, and two of the species that for us are least concerned are for them, incredibly threatened. So we have to really protect what we've got. And why am I bringing this in with temperate rainforests? Well, the fact is that of our 177 international responsibility species, half of them live in temperate rainforests. Hopefully you've all got a chance to be in one of these habitats. They're magical. They're sort of low canopy hazelwoods or beautiful rich oak woods. They're very humid. They have year-round mild temperatures. They're globally rare. 1% of terrestrial land is covered by temperate rainforest. And they're not known for tree diversity. The trees are wonderful because they host epiphytes. So the diversity and the conservation interest lies in the epiphyte communities. If you could get in here with your hand lens, you'd see that that bark is completely covered in a mosaic of lichens. The smooth bark is covered with young smooth bark specialists, and the older stems are covered in mats of bryophytes and older and lichens that require older wood. So the epiphytes can be incredibly diverse in these habitats. Hundreds of species of lichens, hundreds of species of bryophytes in a single small site. And so if we're trying to think about protecting these things and making sure that the epiphytes can manage with all these changes around them, what we want to do is understand the resilience of these things, given the dispersal of the species in there and how the woodlands are managed. So Sally Eaton was a PhD student who worked with funding from Nature Scott with us to try to understand this rainforest system in terms of the dispersal of the species and how they might interact with the characteristics of the woodlands. So what she did was deploy these little stakes. Hopefully you can see there's a stick there with a box on it. And that stick on the box has some little whirly parts on it. And there's air samplers. They're covered in Vaseline. Those go back to the lab and we can amplify up whatever fungi were trapped. So they were deployed in an array from a target tree. So here's the target tree in the middle and from 2 meters to 20 meters out in 4 different directions. And those traps were deployed and those samples were taken repeatedly so we could get quantification of dispersal at different distances from the target trees for 6 different species. And the results of this have been absolutely fascinating. So this is one species, this is one of the target species, Loberia pulmonaria, the lungwort. And this is an example of one of the output models that Sally built. So on the y-axis is the number of trees colonized, going from 0 to 50 at the top. Here we have time on the x-axis from 0 to 200. So we're looking at the number of trees colonized over time. And then the different colors here are the distances that these traps were deployed from the tree. And so what you can see is that there's a real marked difference between these 3 distances and these 3 distances. It will be no surprise to you that these are shorter distances from the tree than these. So at greater than 100 meters down here, the amount of colonization is really low. It takes, we're talking about 100 years to have 2 or 3 trees colonized. But at 100 years, at less than 100 meters distance, then you're looking at 35 to 40 trees colonized. So we can start to actually build a picture of how these species move in the landscape. And this model also incorporates woodland characteristics, extinction probabilities, and colonizations based on repeated surveys and things. So it really gives you a really detailed picture of what these species need to survive in these landscapes. So if you're thinking about planting a new woodland, well, how far away should those trees be? And what kind of inter-tree spacing do we need? So that model was repeated for different species and the same patterns were found under that high management scenario. Under the low management scenario, it just looks bad. So the difference between high and low management scenarios is the tree species that are in the model. So here we have lots of hazels and ashes, which is what the woodlands are like now. But without those hazels and ashes, then the colonization is much, much less. The lichens are much less likely to establish. So it's really exciting to be able to have a model like this where you can actually know how far a species is likely to travel in a given amount of time. So it's a really powerful tool, and Nature Scott was really excited about it. And we hope to, we can roll this out across different sites now to help us understand how to manage for these species that rely on us for their continued existence, which I think is a pretty nice segue into global conservation. I mentioned that I was going to come back to the idea of the IUCN and red listing. So the IUCN is the International Union for the Conservation of Nature. And just like Linnaeus came up with a systematic, I'm not going to put my laser right at him, this way of organizing life, the IUCN has come up with an organized system for assessing vulnerability to extinction. And it's become more and more accepted and more mainstream and more appreciated how important it is for everybody to do the same thing the same way. If we're looking at extinction threat, then we need to look at it in the same way across species groups. And so it hasn't been done very much for fungi, but we're working on it. So this chart shows you the year and the number of species assessed for a red list assessment. So a red list assessment gives you the assessment of vulnerability to extinction. And it's all these classifications I showed you before, from threatened to near threatened to least concern. The first fungi that were assessed were in 2003. There were 2 of them. This is one of them. And not a heck of a lot happened after that, did it? 2 species, 2 species, 2 species. And then something magical happens in 2013. And the thing that was magical in 2013 was that Greg Muller and Anders Dahlberg put together the Global Fungal Red List Initiative. It's kind of a mouthful. It's a GRFLI. It needs a better acronym. No offense, Greg and Anders, it's amazing, but the acronym is a challenge. So what they did was they developed a community-based platform that anybody can sign. You can sign in. You can propose a species, and then you can add data to every single species that you know about. So I can add space. Oh, I know something about it in this country, and I know about it in my country, and you know about it in your country. So people can add the information they have, and they can build a community of people who can start assembling the data, building those threat level assessments for the species. And it's made a massive difference. So it's not just building the assessments, of course, but it's building the community. It's raising the awareness of the need for conservation for fungi. And that's important. So there was a review paper published last year, and there were 597 on that red list at the end. We're near the end of 2022. That was September 2022, and December 22 there's already 625, so it's getting better all the time. And at that point, this is what it looked like. So there were mutualists had been assessed. Those green colors are mutualists, fungi. The life history here of the orange colors are saprotrophs, and then some parasites have been assessed as well. Out of those nearly 600 that were assessed, it's nearly 300 that were threatened. So the other half are least concern or data dependent. And the power of this, of course, is once you have more species, then you can start to pull together trends and understand who's at risk from what. And so on the left, we have the non-lichen fungi and the habitats they're found in, and the lichens on the right. So it's just that power of numbers, isn't it? So we can start to look at the guilds of species and how they're associated with different habitats, the threats, because of course it's not about building the assessment. That's one small part of the assessment. Once we know what those threats are, we can plan what the conservation action should be, and then we can take that action. So that's what that whole cycle is all about. And I think there's a real momentum building for fungal conservation. And I think that's a really exciting trend that's happening around the world right now. So I think there is a future for fungi, and there's a place for everyone. I always like to end a talk like this, inviting everyone to get involved, because even though you might not feel like you're an expert, you might just find that your voice is needed in one way or another to represent, to be your local expert for a county, for whatever. So there's lots of different organizations to find out about, to get involved with. And because of this proliferation of these organizations, that community is building. So I think there is a future for fungi, and I'm excited about it. And I hope that all of you now know something a little bit more about it. That is the end of my talk, and I'd like to just thank Will again for the invitation and all of you for your attention. And I'm happy to take any questions that anybody has.